Hello, my dear students. Today I am going to explain you Chapter 12 Biotechnology and its application, Part 2, Class 12 Subject Biology based on NCERT. But before explaining, I would like to share, and subscribe our YouTube channel so that you may always get an update of new videos. Biotechnological application in medicine. First, we have to discuss what is the importance of biotechnology application in medicine. Biotechnology may have application in animals and human healthcare, either directly or indirectly. So many biotechnology companies that are in top ranking in the world are engaged either in the developing pharmaceutical drugs or in providing services for the human healthcare. The application of biotechnology include that prevention, diagnosis, and cure of disease. At present, we have to know about the 30 recombinant therapeutics have been approved for human use the world over. In India, 12 of these are presently being marketed. Now, the most important thing we have to discuss in this chapter, that is the genetically engineered insulin. Insulin is the smallest protein of the human body and that are very helpful in controlling the blood sugar of the human. So that nowadays so many people are suffering from diabetes, that is type 2 diabetes basically, and in this case the insulin is uh, insulin making is very less, or that are feasible, that are not able to absorb the glucose inside of the cell. So that now we have to discuss about the genetically engineered insulin, and this is totally dependent on the rDNA technology. Naturally occurring molecules which are earlier isolated from the sacrificing animals could be synthesized in cultured bacterial cell, though cloned genes and new drugs could be designed through computer modeling also. Two such drugs are insulin for the treatment of diabetes and one of the interferon, that is treatment against the tumor viruses. So that in this basically insulin is made up of 51 amino acids arranged in two polypeptide chains, A having 21 amino acids and B with 30 amino acids. Basically, my dear students, our body is made up of proteins and proteins is made up of amino acid. So that all the proteins are made up of amino acid. Amino acid is the basic unit of the proteins. So this is the smallest protein of the human body and it has two chains, A having 21 amino acid and B with the 30 amino acids. These two polypeptide chains are interconnected by disulfide bridges or disulfide linkage. So that insulin we have to need that naturally making in our body, but in such cases if the insulin is not made by our body, basically the pancreas having a specific reason, beta cells are responsible for the making of insulin naturally, but in some cases if it is not made, so that the person is suffering from the diabetes or the type 2 diabetes or so we have to discuss how we have to make the genetically engineered insulin. Basically that is a hormone, smallest protein or protein, that is a hormone developed a storage product called proinsulin. Proinsulin has three chains, A, B and C. C chains with 33 amino acid is removed prior to insulin formation. Bacteria cannot be made to synthesize insulin from its gene because it is because be made to synthesize insulin from its gene because of the presence of introns. Bacteria do not possess enzymes for removing introns mediated transcription. In mammals, including humans, insulin is synthesized as a prohormone, like a proenzyme. The prohormone also need to be processed before it becomes a fully mature and functional hormone, which contain an extra stretch called C-peptide. So that the C-peptide is not present in mature insulin. In mature insulin, only two chains are present, A and B. And during maturation into insulin, the C-peptide is removed. The main challenge for the production of insulin using recombinant DNA technology, and this technique was getting insulin assembled into a mature form. In 1983, there is a one American company, that is Eli Lilly, first prepared two DNA sequences corresponding to the A and B chains of human insulin and introduced them in plasmid of E. coli. Plasmid I had already discussed, that is a extra chromosomal circular DNA, double stranded DNA, and this extra chromosomal circular DNA is found in the bacteria. So that here the plasmid of E. coli to produce insulin chain A and B produce separately extracted and combined by creating disulfide bond to form human insulin. So that in this way we have to make the insulin, and that is also known as humulin. Humulin, because that is human insulin. And now we have to discuss about the gene therapy. What is gene therapy? The first question arises, what is gene therapy? It is the technique of genetic engineering to replace a faulty gene by a normal healthy functional gene. Gene therapy is being tried for sickle cell anemia. In sickle cell anemia, the oxygen carrying capacity of the RBC is decreases, and severe combined immunodeficiency, that is also known as SCID, that is a severe combined immunodeficiency. So in this case, the gene therapy, the first case is adenosine deaminase deficiency. This is the first clinical gene therapy was given in 1990 to a 4-year-old girl with ADA deficiency. What is ADA? That is adenosine deaminase enzyme. This enzyme is very important for the immune system to function. It means if the adenosine deaminase deficiency occurs, your immune system is not working properly. And if your immune system is not working properly, then what happened? So many bacteria and viruses are attacking in your body. You are not protected by your own immune system. So that ADA deficiency can lead to a severe combined immunodeficiency. It is caused due to the defect in the gene for the enzyme adenosine deaminase. It means there is a faulty gene which are not able to make the enzyme adenosine deaminase. In some children, ADA deficiency can be caused by bone marrow transplantation. So that this is a for only a few days you have to be a healthy if the bone marrow transplantation. However, in others it can be treated by enzyme replacement therapy. What is enzyme replacement therapy? We have to change the enzyme, but how? In which functional ADA is given to the patient by injection. But in both approaches, the patient are not completely cured because the patient do not have functional T lymphocytes. They cannot produce immune response. because T cells are producing immune response against invading bacteria and viruses. These are the pathogens that are causing diseases in the human being. As a first step towards gene therapy, so that lymphocyte, a kind of white blood cells, are extracted from the bone marrow. When we have to extract the lymphocyte, the kind of white blood cells are extracted from the bone marrow of the patient and are grown in a culture outside the body. A functional ADA, the complementary DNA, using retroviral vector. The retroviral vector, these are the disarmed retroviral vector that is then introduced into these lymphocytes with the complementary DNA, which are re-injected into the patient bone marrow. But these cells do not always remain alive. The patient requires periodic infusion. It means if you have to develop a T lymphocytes or lymphocytes from outside of the body, that is a healthy lymphocytes which are producing the adenosine deaminase enzyme, there is a gene, we have to introduce the gene in the cells of lymphocytes that start producing ADA enzyme. So these are injected into the patient, but these cells are not alive permanently, so that required periodic infusion of such genetically engineered lymphocytes. Genetically engineered, we have to change the faulty gene with the healthy gene in the cells outside of the body. In the cells we have to culture. However, if the isolated gene from the bone marrow cells producing ADA is introduced into the cells at early embryonic stage, it can be permanent cure. So that we have to detect the disease in a embryonic stage, so that we have to introduce these healthy cells at an early embryonic stage. It can be permanent cure. So in this chapter we have to discuss about the genetically engineered insulin and gene therapy, some of the questions that you have to do of your own, or if you have any problem you can ask. And what is the gene therapy? Illustrate using the example of adenosine deaminase deficiency. Define genetically engineered insulin. That I had already discussed, that three chains are there. That is the smallest protein of the human body. That is also one of the important question, important answer of your question. Define genetically engineered insulin. Which famous company first prepared insulin? That you have to do of your own, because I had already discussed in this part rightful form of SCID and ADA, and how many amino acid in insulin, and how many chains of insulin that are working. So these are the this is the part, and these are the questions discussed. My dear students, you have to go through this thoroughly. You read the book also. Thank you, and thank you, students.